It's my pleasure to present to you this work on single particle microscopy of lipid nanoparticles with applications to vaccines and nanomedicines. So why should you be interested in lipid nanoparticles? Lipid, as has been shown by the current pandemic, lipid nanoparticles have shown great promise as a drug delivery vehicle for generic medicines, and in particular, they have proven to be very efficient at producing, responding rapidly to pandemics. However, delivery of these drugs is still a challenge. With the current drug delivery efficiency of the lipid nanoparticles being measured to be around 2%. Hence, we ask the question, how do we improve their design? This can be answered by developing a better understanding of their biophysics. In particular, we are interested in how these nanoparticles behave as a function, as how they respond to changing buffer conditions and the environments in general. Hence, we are interested in how these the structures of these nanoparticles, as well as their loading properties, change as they undergo different environments. We are using lipid nanoparticles formulated in the CLiC lab here at UBC, and these nanoparticles are formed using 4 different lipids, including ionizable lipids, DSPC phospholipids, cholesterol, and PEG lipids. And these nanoparticles are formed through a 2-step process, the first being a rapid mixing process that induces self-assembly of the nanoparticles, resulting in the bilayer structures shown here on the slide. And then the second step involves a dialysis process that changes the buffer to higher pH, and hence resulting in a fusion process that forms the final particle structure. Hence, these nanoparticles are expected to form different structures depending on the buffer they are in, as well as whether they are loaded versus when they are not loaded. Hence, we are interested in understanding these structures at the single particle level. We are using a technique called Convex Lens-induced Confinement. And this technique was developed by my supervisor, Professor Sabrina Leslie. And it involves using a custom device called the CLiC device, shown on this slide. This device is made up of a flow cell, a glass flow cell that's made up of 2 cover slips with inlets on both ends, and a spacer tape that forms the chamber of the flow cell. And then one of the surfaces of the flow cell can be fabricated with micro features which allow us to trap different molecules of particles in the surface. We trap these particles by deforming the flow cell by pressing down on one of the surfaces to deflect it into the other surface. And we achieve this by using the pusher lens shown again in the image of the device. The pusher lens is controlled by a piezo controller that allows us very high precision and control. In particular, for lipid nanoparticles, we use the CLiC technique to image single particles that are diffusing confined in microwells. This is achieved by initially loading the flow cells with lipid nanoparticles in solution. Then by gently pushing down on the top cover slip, we can trap the nanoparticles in these microwells as shown by the bottom insert. And then by trapping these nanoparticles at a single particle concentration, we can then perform single particle tracking on these trapped particles, which are free diffusing. To perform the particle tracking, we fit a 2D Gaussian to the intensity distribution of the particles. And from this fitting, we can get a very precise localization of the nanoparticles as shown by this movie, and hence generate a long trajectory of a freely diffusing particle that is trapped in the well. And from this particle tracking, we get both the position information as well as fluorescence signal information of this particle. From the position information, we use MSD curves, which we use diffusion models. We fit diffusion models to the MSD curves to get diffusivity. The diffusivity directly tells us what the particle size is, while the intensity information, from the intensity information, we can actually count how many dyes you have per particle. So for example, if you label the drugs with a dye, a fluorescent dye, then perform photo breaching measurements, you can count how many dyes you have or how many drugs you have in a single particle. Hence, from these measurements, we get both size and intensity or loading per particle. And by performing this on thousands of particles, we can then build distributions of size and distribution of intensity from single particle measurements. And since these are from single particle measurements and coming from the same particles, we can then correlate the two distributions to get information about structure of these nanoparticles. Hence, we apply this method to study how the structure of these nanoparticles changes as a function of changing buffer conditions. So hence, the first set of results in this study, we looked at how particle structure changes as a function of pH. Again, as mentioned, we track single particles and then build distribution of size and loading of those particles. For this particular study, we looked at unloaded lipid nanoparticles, and we labeled these nanoparticles with a lipophilic dye. These dyes have a hydrophilic head with hydrophobic tails. Hence, they can only insert themselves in the surface of the particle. They don't like to be in the volume of these particles. Hence, from these measurements, we can then get a distribution which is dependent on the arrangement of these dyes on the particle. By correlating the size and intensity distribution, we can directly measure how these dyes are distributed on the particle. In particular, here by taking the log of the distribution in size and the log of distribution in intensity, and correlating the two distributions, the scaling of the scatter plot, the resulting scatter plot gives us a scaling of intensity with size. Here we are getting a scaling of 2, which basically tells us that the dyes are arranging themselves as a shell-like structure, which is in agreement with the distribution of dyes on the surface of the particle. Next, we applied the same method to look at lipid nanoparticles that have a bilayer structure. In this case, some of the dyes are actually on the inner surface of the bilayer, hence they are no longer following a shell-like structure, but hence we do expect a more volume-like feeling because some of the dyes are actually on the inner surface of the bilayer. And by correlating size and intensity distribution again, we get a scaling of 2.3, and we performed statistical test to verify that these two distributions are statistically different. Hence, our measurements are sensitive to the dyes being inside of the particle. These measurements are in agreement with cryo-EM measurements, which show bilayer structures in the low pH condition with oil droplet structure in the high pH condition. Next, we apply these measurements to study how lipid nanoparticles the drugs in the lipid nanoparticles are distributed. In this case, we label the drugs, specifically siRNA drugs, with fluorescent dyes. And by looking at how the scaling of the intensity of these dyes scale with the size of the particle, we can get information about how they arrange themselves inside of the particle. Again, we get single particle measurements, build distributions of size and loading. And then, as mentioned again, we expect a shell-like structure if the dyes, if the drugs are arranging themselves as a shell in the particle, while we expect a volume-like scaling of 3 on the log scaling plot if the dyes are fully distributed in the particle. However, both of these models don't fit to our data well. This can be explained by the dye distribution or the drug distribution being more complicated, basically having nugget-like structures in the particles, whereby the intensity, the loading number and size of the particle are not correlated as well as the two models are showing. I would like to end by thanking everyone in the Leslie lab, especially my supervisor, Professor Sabrina Leslie, as well as our collaborators. I would also like to thank Yife Gu, who contributed to this work, as well as Dr. Romain Betty, who also contributed to this work. I would like to end by summarizing the results. In these measurements, we basically developed a technique for single particle imaging of lipid nanoparticles. We apply this technique to measure size and loading of these nanoparticles. And from the resulting distributions, by correlating those results, we get structural information of these nanoparticles. Thank you for listening to my talk, and I'll be happy to take any questions.
Great, thank you very much, Albert. I am pleased to open the floor to questions from the audience. Please either submit a written question or raise your hand. And while everyone is typing, I will ask a quick question of my own. Albert, how do you make sure that the surface interactions aren't affecting your measurements?
Yes, that's a very good question, actually, because so these nanoparticles are supposed to be neutral, but they actually do have a positive charge, and the glass surface is negatively charged when cleaned. Hence, we perform surface passivations on the flow cell surfaces to ensure that the particles don't stick. And then we also perform a series of measurements where we look at size measurements as a function of well depth to ensure that we are far enough from the surface that the surface interaction are not affecting our measurements.
Great, thank you. Thanks. Marshall, are there any hands or questions?
I see neither at present.
Okay, I'll take this chance to ask another question. Albert, could this technique be applied to look at dynamic changes in LNPs, such as when you're changing pH or other buffer conditions?
Yes, so the results I just showed, we are using particles that are trapped in microwells that are fully sealed because the two surfaces completely come into contact. But we also have devices where we leave some spacing between the two cover slips by basically placing pillars on the other surface. And also having a microchannel allows us to change, for example, buffers or send in reagents and watch as the lipid nanoparticles are responding to the changes.
Great, thank you. Marshall, any questions?
Yep, we have a written question I'll read out. Oh, actually two. Starting with the first, which is, what is the size measurement range that you can reliably work with?
Yes, that's a very good question. So it depends on how fast you can track the particle. So the range of sizes we've looked at are tens, like 10 nanometer to hundreds of nanometers. So yeah, so at the lower end, you want to image fast enough. So the limit will be how fast your camera can go. While at the higher end, it's the depth of your well. So you want the wells to be deep enough that you don't feel the surface effects. But if they are too deep, then it's a question of whether your objective can keep the sample in the imaging plane. Yeah, so I would say tens to hundreds of nanometers. Yes.
Great, thank you. The second question comes from Ariadne Tuckmantle-Lido again, who wrote, amazing talk. I was wondering how hard it is to apply this CLiC method to get single particles, and if that would work with other particles, like gold nanoparticles. This may be very interesting for Raman studies that struggle to get single particles for analysis.
Yes, so that would be really interesting. So the work I'm showing is fluorescently labeled lipid nanoparticles. Working with label-free is something we're interested in. So definitely, so we've looked at quantum dots, but gold nanoparticles will probably need, yeah, you have to go into label-free microscopy. So that's something we're interested in expanding into.